Hi everyone, welcome to your Clean Neat English batch. Good afternoon, how are you all doing? Good afternoon, Navya, Madhu, Tiso, Sneha, Sharitra, Tulsi, Akshara, Riya, Bhumika, Pratibha, Anam, Simran, Suma, Nikita, Pratik, Shravani and everybody else. Vanakkam. Okay, so how was your weekend? I think on Saturdays you have like one class for 1.5 hours. Did you all use your weekend productively? In the last 48 hours, did you study for at least 10 hours? How many of you used it productively? How many of you wasted time and regretted it later? Did you all study at all? Or you just relaxed and re-energized? Hi Suma, ನಾನು ಆರಾಮಿದ್ದೀನಿ. Thank you. Just remember, the amount of effort you put in, the results that you're going to get is going to be proportionate to it. If you're doing the same thing you did in the last attempt, the results are going to be the same. If you want newer results, then you have to, upgrade yourself. Okay? 8 hours only. Not enough, not enough. All right. So in our last class, we had completed the light reaction of photosynthesis. Does anybody have any doubts from the last class? Some of you had asked me doubts over Telegram, which I will clarify now. One doubt that I frequently got was whether photosynthesis occurs at the night, whether oxygen is released by plants at night. The answer is no. The reason is at night usually the stomata are closed, and also for photosynthesis to take place, light is an important requirement. If there's any artificial light that the plant is receiving in the form of street lights or something, then probably it can photosynthesize, but otherwise it does not. Doesn't matter, because so much oxygen will be released during the day that will still be present in the atmosphere that it will suffice. All right. And another question that I got was with respect to LHC complex. LHC complex is light harvesting complex. So according to NCERT, all the antenna molecules are called the light harvesting complex, and in the reaction center you will have chlorophyll A either 680 or 700. Another doubt that I got was during cyclic photophosphorylation. I think Siddhika asked this doubt. During cyclic photophosphorylation, does chemiosmotic hypothesis take place? Does the gradient, proton gradient, form across the thylakoid membrane? Yes, even during cyclic photophosphorylation, we saw how ATP is getting synthesized. If ATP has to get synthesized, then you need that proton motive force. Where does that proton motive force come from? Because of the gradient, right? So wherever ATP is getting synthesized during photosynthesis, if photophosphorylation is happening, then obviously a proton gradient will exist. Okay? Apart from this, does anybody else have any other doubts? Oh yeah, also about the forms that you all had submitted, I got a lot of questions on when the session will be conducted. I had told you previously that I will try to conduct it on Saturday, but the problem, I'll tell you what, is that I've got like so many responses and the concerns are so diverse. Like different people, one major thing that most of you are struggling with is mobile phone addiction. But apart from that, there are so many diverse concerns that I can't put it all together into one session and just do it for the sake of it. I have to make it meaningful. Even if you're watching that session, your strategy should improve, your preparation should improve at least by 1%, otherwise it will not make sense. So I'm planning different sessions for different concerns instead of just combining everything and making a khichdi out of it and pushing it out to you all. Okay, so that is why it's taking time. Hopefully, as soon as possible, hopefully in the coming weekend, I will try and design it for one or the other concern. Hopefully for how you can overcome mobile addiction, because most of you are, most of you resonate with that because most of you are facing it. So we'll take it one step at a time. Like each week we can have a session for a different concern instead of taking one 4-hour session, 3-hour session and dumping everything on you. Okay? Okay. So, Tulsi is asking for the difference between oxidative phosphorylation and photophosphorylation. Right now, I've only explained to you photophosphorylation, which is what happens in photosynthesis during ATP production. That is photophosphorylation. I can't tell you about oxidative phosphorylation right now because we've not covered that yet. That will come in our next chapter, respiration in plants. Over there, I will highlight the difference between what happens in photo and what happens in oxidative. There's another type of phosphorylation called substrate level phosphorylation. That also we will cover in our next chapter. For now, just understand photophosphorylation. Okay? Alright, so if everyone's ready, we'll get started. Give me a thumbs up. Sonu, who are you calling Choco? What is Choco now? No, Bindu, I will not do PYQs because PYQs I've already done after each chapter in the class itself. What I will do after we finish 11th grade syllabus is, see, this is one chapter. This chapter we'll finish this week. In another probably 2.5 weeks, we'll finish the 11th grade portions. After that, I will have a quiz for all 11th grade chapters, but it will not be PYQs. It will be my own questions. This time I will do proper, rechecking and get the questions. I will not repeat the questions. And PYQs, I will give you a document for each chapter. I will give you a PDF where you have questions. You can revise that frequently. Okay. Again, I will not conduct a quiz for PYQs because after each chapter we are doing it anyway. All right. Since I can see a lot of thumbs ups, we'll get started. What are we going to learn today? We're going to learn about the dark reaction. And then we will learn about a process called photorespiration. Okay, it's interesting, but it can seem a little tricky. So pay attention, don't get distracted. As always, the same instruction. So what do we know till now? Let's do like a quick recap. What did we learn in the last class? We learned about the light reaction. Light reaction. In the light reaction, what happens? You have sunlight that chlorophyll will absorb, and the energy that the sunlight has is used to make molecules called ATP and NADPH. While this is happening, photolysis of water will take place. Water will split to give us oxygen, electrons and protons. This synthesis of ATP is called photophosphorylation. This is of two types, cyclic and non-cyclic. In cyclic, you only have PS1. In non-cyclic, you have PS1 and PS2, right? In cyclic, only ATP is produced. In non-cyclic, ATP plus NADPH plus H plus is produced. Over here, water will split to give rise to oxygen, which doesn't happen here. Okay, so this is something that we already know. What we're going to learn today is how for carbon fixation, when we say carbon fixation or carbon dioxide assimilation or carbon dioxide fixation, what we essentially mean is carbon dioxide is an inorganic molecule. It's gaseous, it's present in your atmosphere. Plants take that up and convert it into an organic molecule. So that process is called carbon dioxide assimilation or carbon fixation or carbon dioxide fixation. That is what happens in the dark reaction. Alright, so we will learn about how dark reaction occurs today. So what is dark reaction? It has alternate names. Dark reaction, like I've told you before, is a misnomer. It is an incorrect name because dark reaction does not require darkness. It does not happen in the dark. It's called dark reaction just because it does not directly depend on the sunlight. Okay. So what are the alternate names for dark reaction? You can call it light independent reaction. It happens during the day, simultaneous with the light reaction, but it does not directly depend on the light to occur. What does it depend on then? It depends on the products of light reaction. What are the products of light reaction? The products of light reaction are ATP and NADPH. I just told you that. So for carbon dioxide fixation during light reaction, during dark reaction, those products are utilized. Okay, so we can call it dark reaction. You can call it light independent reaction. You can also call it biosynthetic pathway. In photosynthesis, we are synthesizing something. What are we synthesizing? We are synthesizing glucose. Where exactly does that synthesis of glucose happen? It happens during the dark reaction. Inside living organisms called plants. Bio is living. So, biosynthetic phase. There, in this phase is when actually glucose is getting formed. Alright, so that's why it's called the biosynthetic phase. Remember all of these three terms. Any of them can be used in the question. You should know biosynthetic means dark reaction. Photochemical means light reaction. Alright, now like I already mentioned, this does not directly depend on the presence of light. It depends on its products, ATP and NADPH. Yeah, that's about it. How do we get to know about the dark reaction? This was around the time when World War II was happening and most people were using radioactivity to make nuclear bombs and destroy countries and kill people and all of that. Around the same time, a genius scientist called Melvin Calvin, this Calvin has C, your Kelvin in physics has K, right? So this is Melvin Calvin. He used the same radioactivity, the same principles of radioactivity to do something more useful, something more meaningful. So what he basically did was, we are trying to understand how exactly carbon dioxide fixation takes place. We know from our early experiments, previous experiments we studied in the first class, that carbon dioxide is taken into the plant, it makes glucose, it stores it as starch, it releases oxygen, all of that we know. How exactly is this carbon dioxide going to form glucose? Is it a direct process? Are there any intermediates? If there are intermediates, which are those? Is it a straight linear pathway or is it a cyclic pathway? These are some things that they were researching about. So what he uses is a very simple photosynthetic organism called chlorella. Everybody knows about chlorella, right? It's a unicellular eukaryotic photosynthetic organism. So he was studying photosynthesis in chlorella. What he does is, for plants to perform photosynthesis, carbon dioxide is required. The carbon dioxide that he provides to this chlorella was radioactive. The carbon dioxide had radioactive carbon, which is C14. Are you getting my point? See, he is growing chlorella. These are chlorella green algae, unicellular green algae. For this to undergo photosynthesis, it requires a source of carbon dioxide. The source of carbon dioxide that he is providing, he and his colleagues were providing to these chlorella cells was a carbon dioxide that had C14 carbon, which is a radioactive carbon. The only carbon dioxide these organisms are getting is this radioactive carbon containing carbon dioxide. So if they have to make glucose, then obviously they will use the same C14 carbon, right? So it takes it up into its cells, and inside the cells, whatever reactions happen, whatever intermediates will form, those should ideally have this C14 carbon. If they are carbon containing compounds forming as intermediates, they should ideally have this C14 carbon. So what they do is, at frequent intervals, they will isolate whatever chemicals are present in the chloroplast of these cells. Okay, so they know for sure. Remember, we learned that there are special bodies called chloroplasts. Inside that, chlorophyll pigment is present. In these special bodies, glucose is formed. It is stored as starch. We've learned about it. Julius von Sachs. So what we know already is that inside the chloroplast is where photosynthesis is taking place. So what they will do is, at regular intervals, they will take out cells, they will remove isolate the chemicals that are present in the chloroplast and check for which compounds there have C14. So after the first few minutes, they will get one compound. After the next few minutes, probably they will get another compound. Based on the compounds that they obtained, they kind of traced the entire pathway. Okay, so for this, they were, I think, sometime in 1959, 1960, around that time, they were even awarded with the Nobel Prize. This is called a Calvin cycle. The dark reaction is also called Calvin cycle because Calvin was the main head researcher who was doing this project, who was part of this research project. Of course, with him, a lot of co-workers were there, but he was awarded the Nobel Prize for his discovery. So based on Melvin Calvin's work, we found out that the first stable product to form during dark reaction was a 3-carbon organic acid. It was a 3-carbon compound, and we call it 3-phosphoglyceric acid. Alright. So Melvin Calvin used radioactive C14 to synthesize, to study algal photosynthesis. Okay, they've not mentioned which alga it is in your book, so don't worry about chlorella or not, or chlorella or which other species. If it's not mentioned, take it out of your head. You already have sufficient examples to remember, so don't worry about the name of this. Next one. Their studies led to the discovery that the first carbon dioxide fixation product was a 3-carbon organic acid. What does that mean? Carbon dioxide will directly not become glucose. From carbon dioxide, you're directly not getting a 6-carbon glucose. What you're getting first contains only 3 carbons. It is an organic molecule. It is an organic acid. The name of that is 3-phosphoglyceric acid. In short, you can call it 3-PGA. Now, why are we calling it 3-PGA? This is a 3-carbon containing molecule. So, you have 3 carbons. If you're labeling 1, 2, 3 carbons, to the 3rd carbon, a phosphate group is attached. Okay, that's why it's called 3-phosphoglyceric acid. 3-phosphoglyceric acid does not mean 3 molecules of phosphoglyceric acid are forming. The name of this molecule itself is 3-phosphoglyceric acid. Okay, to the 3rd carbon of this compound, a phosphate group is attached. Alright, I repeat. They were studying photosynthesis in algae. They used radioactive isotope of carbon, which is C14. They supplied only carbon dioxide containing the radioactive isotope of carbon to the algae that they were studying. At frequent intervals, they checked for different chemicals present in the chloroplast. By doing all of these experiments, they realized that the first product of carbon dioxide fixation is a 3-carbon organic acid. Its name is 3-phosphoglyceric acid. Okay, so this is the first thing that they found out. Of course, they worked out the entire cycle, about which we'll study later. Alright, now similar to whatever experiments Calvin had conducted using chlorella, people started conducting experiments using different plants across different parts of the world. They did the same kind of experiment using radioactive carbon, but they did it on different kinds of plants. When they did the same experiments on different plants, what they noticed was that in some plants, the first product that was formed was not a 3-carbon compound, it was a 4-carbon compound. It was an organic acid itself, but it did not contain 3 carbons, instead it contained 4 carbons. And that 4-carbon compound was oxaloacetic acid. In short, you can call it OAA. So, depending upon whether during carbon dioxide fixation, the first product that is forming is of 3-carbon or 4-carbon, you have 2 different pathways. If it contains 3-carbon, if it is 3-phosphoglyceric acid, then such plants are called C3 plants. Such plants where the first stable compound formed contains 3 carbons, those plants, that group of plants are called C3 plants. And the kind of pathway there is called C3 pathway. And in plants where the first stable compound formed is a 4-carbon organic acid, oxaloacetic acid, those plants are called C4 plants, and the pathway by which carbon dioxide assimilation happens there is called the C4 pathway. If C4 pathway is happening, they are C4 plants. If C3 pathway is happening, they are C3 plants. Majority of plants are C3 plants. You have comparatively fewer species of C4 plants. What is the advantage? Which one is better? All of that we'll study in tomorrow's class. Okay. So carbon dioxide assimilation can happen in 2 ways. The first product, if it is a C3 acid, which is PGA, then you call it C3 pathway. If it is a 4-carbon acid, oxaloacetic acid, then we would call it the C4 pathway. Simple, right? Today we will learn only about C3 pathway. In tomorrow's class, I will teach you about C4 pathway. Up until this point, does anybody have any doubts? Let me know. Okay, cool. So, Nida is asking if 7 hours of sleep is sufficient. 7 hours of sleep is sufficient as long as there is no interruption in between. Like, you should not wake up in the middle, check your phone for 20 minutes and then go back to sleep. So if there is no interruption and you're getting like a good quality 7 hours of sleep, then it is sufficient. Hi, Samaira. Welcome to the class. All right, let's move on. Okay. I'll talk about how you can improve your sleep quality, how you can manage your time, all of that in that session. I have all of these points in mind. Now the challenge is for me to fit it all together in that one session. Yeah, I'm working on it. I'll get like the best session for you as soon as possible. Okay? Cool. Now, yeah. See, there's this thing that Tawfiq, yes, you have to remember the whole pathway of C3 pathway, whatever is there in NCERT, just that much. It's not really difficult. It's pretty simple. Okay. So, yeah, this is called C subscript 3 pathway. So what happened was, you know that carbon dioxide is getting fixed, right? So this carbon dioxide contains one carbon. This carbon dioxide combines with some other molecule inside the chloroplast. Let's call it molecule A. We don't know which one that is right now. As a product of this carbon dioxide fixation, we know from Calvin's experiment that we receive the first product. The first product, pay attention and listen, don't get distracted, product which is 3-PGA. Now this 3-PGA contains 3 carbons. What would be the automatic assumption? What would be the automatic first line of thinking? Carbon dioxide contains 1 carbon. It's combining with some other molecule and forming a 3-carbon compound. So how many carbons would this compound possibly have? They thought that the molecule that is accepting the carbon dioxide molecule, the primary carbon dioxide acceptor here, was a 2-carbon compound. So 1 carbon, 2 carbon combining together giving a 3-carbon compound. And for a really long time they kept on looking for that 2-carbon compound. But turns out the primary carbon dioxide acceptor in C3 cycle is not a 2-carbon compound, it's a 5-carbon compound. If it's a 5-carbon compound, you have 1 carbon here, then you should get a 6-carbon compound as a first product, right? Then how is it that we're getting a 3-carbon compound? I'll explain that to you. Okay. Later, after several years of studies and research, they got to know that it is not a 2-carbon compound, it is a 5-carbon compound, and this has a very nice name. It's called RUBP. Okay, this 5-carbon compound is called RUBP. It is ribulose bisphosphate. RUBP is a short form for ribulose bisphosphate. Why are we calling it bisphosphate? Because it has 2 phosphates. This, like I already mentioned, is a 5-carbon ketose sugar. Actually, in sugars, carbohydrates, monosaccharides, you will have, you call it like triose, tetrose, pentose, like that, right? So 5-carbon means pentose. What does ketose mean? Depending upon whether it has an aldehyde group or a ketone functional group, you will call it ketose or aldose, but that is beyond the scope of your syllabus. You might probably learn, no, it's not even there in biomolecules for 11th grade. So don't worry about it. Just remember that RUBP is a ketose sugar. All right. So yeah. So this 5-carbon RUBP obviously will have 5 carbons, and to the first carbon a phosphate group is attached, and to the 5th carbon as well a phosphate group is attached. Because it's attached to 2 different carbons, we call it ribulose 1,5 bisphosphate. Why can I not call it diphosphate? Phosphate. ADP also has 2 phosphates. ADP, adenosine diphosphate. Over there I'm calling it diphosphate. Why am I calling it bisphosphate here? That's because the 2 phosphates are attached to 2 different carbons. Over there, to the same carbon, you will have, to the 5th carbon of that nucleotide, you will have 2 phosphates attached. But over here, to 2 different carbons, you have 2 phosphates attached. So we call it ribulose bisphosphate. So far, we know about the first stable product that is formed, which is a 3-carbon containing compound, 3-phosphoglyceric acid. We know that the primary acceptor, we call this the primary acceptor, okay, is a 5-carbon ketose sugar. Its name is RUBP, ribulose bisphosphate. Alright, now let's see how this pathway works. This operates in a cyclic manner. I told you that Melvin Calvin and his colleagues worked on deciphering, finding out, decoding how exactly, what is wrong with you, dude? How exactly it works, right? How exactly it functions. So they found out that this occurs in a cyclic manner. That means something is getting regenerated, right? So what was getting regenerated? If it's happening cyclically, that means some molecule is getting formed again, right? What is getting regenerated is the RUBP itself. This has 3 steps. The first step is called carboxylation. The second step is called reduction, and the third step is called regeneration. First step, carboxylation. Second step, reduction. Third step, regeneration. What is getting regenerated? We already know. We know that RUBP is getting regenerated. Carboxylation, what is happening? That also we know. RUBP is only getting carboxylated. Carboxylation means what? Carboxylation means addition of carbon dioxide. To which molecule addition of carbon dioxide is happening? To RUBP. Right. So, carboxylation is happening to RUBP. It's forming this product. We already know that. In another step, regeneration is happening. Regeneration is also of RUBP. So, these are the 3 steps in order. Carboxylation, reduction, regeneration. So, let's learn about how exactly this pathway functions. Pay attention, don't get distracted. So right now, we will start with the first step. What is the primary acceptor? It is RUBP, right? Okay, let me write it here. All right. So we're starting with RUBP. This is how it's written. Capital R, lowercase u, capital B, capital P. Now this RUBP contains 5 carbons. To this RUBP, a molecule of carbon dioxide will get added. How much carbons do carbon dioxide have? It has 1 carbon. What happens during a dark reaction or C3 pathway or Calvin cycle is this 5-carbon compound combines with 1-carbon compound to form a 6-carbon compound, but it's so stable that immediately after it's formed, it will split and form 2 molecules of a 3-carbon compound, which is 3-phosphoglyceric acid. Now this 3-phosphoglyceric acid also contains 3 carbons. I have here 1 molecule of RUBP. I have here 1 molecule of carbon dioxide. 5 plus 1, 6. I am forming 2 molecules of 3-phosphoglyceric acid. First a 6-carbon compound forms, but it's so unstable it splits immediately into 2 molecules of a 3-carbon compound, which is phosphoglyceric acid. This is our first step, and this step is called carboxylation. Now for carboxylation to occur, an enzyme is required. An enzyme catalyzes this process of carboxylation, and that is called Rubisco. Catalyzed by an enzyme called, again C, R capital U BISCO. What is the full form of Rubisco? It's called ribulose bisphosphate carboxylase oxygenase. How are enzymes named? Enzymes are named based on which substrate they are acting on and what function they are performing. Over here we are calling this enzyme Rubisco, ribulose bisphosphate, which is the substrate, carboxylase oxygenase, which is the function of this enzyme. So what happens in this Rubisco is, this is like any other enzyme. It will have its active site. This will have two active sites. Okay. To one active site, it will bind to RUBP all the time. There is no change in this. It's like if I am Rubisco, on to my right hand, I will always hold on to RUBP molecule. But my left hand can either pick up carbon dioxide or pick up oxygen. If I am picking up carbon dioxide or oxygen, regardless of what I pick up, I will take that and I will combine it with RUBP. Okay, RUBP is fixed in one hand. On the other hand, depending upon certain conditions, I can either pick up oxygen or I can pick up carbon dioxide. If I pick up oxygen, I will add it to RUBP. Even if I pick up carbon dioxide, I will add it to RUBP. If I am picking up carbon dioxide and adding it to RUBP, that is my carboxylase activity. If I am picking up oxygen and adding it to RUBP, that's my oxygenase activity. During Calvin cycle, what happens is its carboxylase activity. Right now, this enzyme here is combining carbon dioxide with RUBP. So, it is showing its carboxylase activity. So, over here, this can either be oxygen or it can be carbon dioxide. Over here it is carbon dioxide. So you can call this enzyme RUBP carboxylase. Okay, in Calvin cycle, in the dark reaction, it is showing its carboxylase activity. But it can show oxygenase activity also. When it will show that, I will explain it later. For now, what do we know? We have one 5-carbon compound RUBP. We have one carbon compound carbon dioxide. We have one molecule of each. Both of them are getting combined. So RUBP is getting carboxylated. We call this step carboxylation. Instead of forming one big 6-carbon containing molecule, it forms 2 3-carbon containing molecules, which is 3-phosphoglyceric acid. Alright, 3-PGA. This step is called carboxylation. Now, our next step is reduction. The second step is reduction. During reduction, this 3-phosphoglyceric acid is going to get converted into an aldehyde. That aldehyde is called It is called 3-phosphoglyceraldehyde. It's called 3-phosphoglyceraldehyde. In your textbook, this is given as triose phosphate. Triose means it has 3 carbons and 1 phosphate, so it's called triose phosphate. This also contains 3 carbons. We have 2 molecules of 3-phosphoglyceraldehyde. Acid to aldehyde you've studied in chemistry, it is reduction, right? Now what happens here? is, I told you that for the light reaction to take place, for the dark reaction to take place, we need ATP and we need NADPH. In this step, ATP and NADPH will get used. For each molecule of phosphoglyceric acid getting reduced to phosphoglyceraldehyde, PGAL, we require one molecule of ATP, one molecule of NADPH. Right now, there are two molecules. So over here, I will need two ATPs and two NADPH. Okay. So in this reduction step, ATP and NADPH will get used up. How many ATP? 2 ATP plus 2 NADPH will get reduced and form it will form 2 ADP plus inorganic phosphate plus 2 NADP plus. As phosphoglyceric acid is getting reduced to phosphoglyceraldehyde, NADPH will get oxidized to NADP plus. Carbon dioxide does not have hydrogen. Glucose has hydrogen. What is giving hydrogen to glucose? It is coming from NADPH. Where did the NADPH get its electron from? If you think about it, where did NADPH get its electron from? NADPH is formed only in non-cyclic photophosphorylation. NADPH gets its electron from PS1, right? But if you think about it, water split and gave electron to PS2. PS2 gave its electron to PS1. PS1 ultimately gave its electron to NADP+. So if you think about it, the electron that NADPH has actually came from water molecule, right? The water molecule split and that electron itself is being circulated throughout. So the electron from the water molecule is now with NADPH. That electron and that H is what get used during reduction process. So in reduction process, oxidation is also happening, reduction is also happening. What is getting reduced? 3-phosphoglyceric acid is getting reduced. Reduced to what? 3-phosphoglyceraldehyde, acid to aldehyde. Simultaneously, oxidation is happening of NADPH and that is getting oxidized to NADP+. Alright, so over here in the reduction step, per molecule, you need one ATP, one NADPH because we have two molecules, we are making use of two ATPs and two NADPH. Alright, over here I have two molecules of 3-carbon compound. Alright, now in the next step, which is called regeneration, what happens is in this step called regeneration, just give me 1 minute. Yeah. So in this step called regeneration, 5-carbon RUBP will be regenerated. Over here, how many carbons do we have? We have 2 molecules of a 3-carbon compound. So totally we have 6 carbons here. 2 molecules, 3-carbon compound, we have 6 molecules here. Of these 6 molecules, 6 carbon atoms. Okay, let me use this color. 5 carbons will be used up from this to regenerate RUBP. During this process, another ATP will be used up. So of these total 6 carbon, of these total 6 carbon, 5 are utilized in the process of regenerating RUBP. What is left? We have 1 carbon left. The 1 carbon will be withdrawn from this cycle. That 1 carbon will be withdrawn from the cycle and used in the formation of glucose. This glucose contains 6 carbons. For the formation of glucose, we need 6 carbons. But right now, per cycle, we have withdrawn only 1 carbon from the cycle. So if we have to synthesize 1 molecule of glucose, the same cycle has to go on for 6 times. After this cycle has gone on for 6 times, we would have withdrawn 6 carbons from this cycle. That can be used for the synthesis of glucose. And this glucose, if it has to be transported, it will be transported in the form of sucrose. If it has to be stored, it will be stored in the form of starch. We started off with RUBP that contains 5 carbons. RUBP got carboxylated with the help of the enzyme Rubisco, and it formed 2 molecules of a 3 carbon compound, which is phosphoglyceric acid. You can call it 3-phosphoglyceric acid. In this step, neither ATP nor NADPH is used up. In the next step, the 3-phosphoglyceric acid will get reduced to 3-phosphoglyceraldehyde. As reduction of 3-PGA is happening, simultaneously oxidation of NADPH will happen to NADP+. In this reaction, ATP also will be used. How many ATP, how many NADPH? 2 ATPs, 2 NADPH. Per molecule, 1 ATP, 1 NADPH. Because we have 2 molecules here, 2 ATP, 2 NADPH is used. This step is called reduction. Now at the end of reduction, what do we have? We have 2 molecules of 3-phosphoglyceraldehyde. Since we have 2 molecules of a 3 carbon compound, you totally have 6 carbons. Of these 6 carbons, 5 carbons go back and regenerate RUBP. This is actually an over super simplified form of Calvin cycle. Calvin cycle is really very complicated. In regeneration, it is a multi-step reaction. In reduction, it is a multi-step reaction. Over here, you have fructose, pseudoheptulose, xylulose, erythrose, different carbon compounds coming, but that is not given in your syllabus. Though I am showing it as one arrow, this is not really one arrow. This is like a lot of arrows. Many intermediates will form here. Even this is not a single arrow, but for your grade, for your syllabus, this is how it is. Alright, but here, remember, it's not as carbons are getting regenerated to form RUBP, many intermediates will be formed here. So in that process, ATP will be used. 1 ATP will be used in the process of regeneration. Here, this cycle will go on for 6 times. After 6 times, 6 carbons have been withdrawn from the cycle. Those 6 carbons will be used in the formation of glucose. If glucose has to be stored, it's stored in the form of starch. If it has to be transported, it's transported in the form of sucrose. Alright? If you have any doubts, let me know. Sudha is asking if Calvin cycle and C3 are both the same. Yes, it is. There's a video lag, or am I talking really slowly? Ayyo, okay, I will explain again. When did the video lag? I didn't notice at all. No, the video didn't lag. I went to drink water. I took a break. Water break. Are you able to see it clearly here, clearly now? Is it lagging even right now? Okay. So what basically happens during regeneration is making another molecule, making the same molecule again, regenerating it. How are we regenerating it? Over here we will have 6 carbons. You will remove 5 carbons from here and make a 6 carbon. Make a 5 carbon RUBP. 1 carbon will be pending, that is utilized in the formation of glucose. You withdraw it from this cycle and use it in the formation of glucose. Now, per cycle of Calvin cycle, per turn of Calvin cycle, how many ATP and how many NADPH are used? Per turn of Calvin cycle, this actually has to take 6 turns to form 1 glucose. Per turn, how many will be used? Totally 2 here, 1 here, 3 ATP will be used, 2 NADPH will be used. So, if you look at it, what goes in the Calvin cycle? Per carbon dioxide, or let's say per turn. This is per turn or per carbon dioxide molecule. What happens? What goes in? 1 carbon dioxide goes in. How many ATP are utilized? 2 ATPs are utilized. How many NADPH is utilized? 3 ATP and carbon dioxide. 3 ATP and 2 NADPH is utilized. Again, per turn, 1/6 of a glucose molecule is formed. 3 ADP are formed. 2 NADP plus is formed. They can ask you questions based on ATP and NADPH, whether it is per carbon dioxide, whether it is per turn or per glucose molecule. That is something that you have to pay attention to. If they are asking it per turn or per carbon dioxide, then these are the numbers. If they are asking for the formation of 1 glucose molecule, this is for 1 glucose molecule. For 1 glucose molecule to happen, this should happen 6 times, right? It should happen in 6 turns, only then 1 glucose molecule will form. So in that case, 6 carbon dioxide is used, 1 glucose is formed, 18 ATPs are used, 18 ADPs are formed, 12 NADPH is used and 12 NADP plus is formed. Let's try to understand it like this. Okay. See, we start off with RUBP, right? So we have RUBP here. RUBP has 5 carbons. I am going to take 6 molecules of RUBP. So I am going to have a total of 6 5 times is how much? 6 5 times is 30. RUBP 6 5 times is 30. So over here I will have 30 carbons, right? To this I will add 6 molecules of carbon dioxide. So over here I have 6 carbons. From this, what do I get? I'm only showing carbons here so that you understand. I'm not writing the other details. So from here we will get 3 phosphoglyceric acid. How many do we get? We will get 12 molecules of 3 phosphoglyceric acid. Each one will have 3 carbons. 3 12 times is 36. So I have 36 carbons here. This will get reduced next. When it gets reduced, I will have 12 molecules of 3-phosphoglyceraldehyde. Since there is no carbon dioxide added or removed here, the number of carbons will remain the same. It will be 36 carbon. of these 30 carbons, 36 carbons, 30 carbons are used in the regeneration of RuBP, 6 molecules of RuBP. So 30 carbons is how much we started with. 30 carbons is what we are withdrawing to regenerate this. If you take out 30 from 36, how much has remained? 6 is remained. 6 remains, right? So this 6 carbon is used in the formation of C6H12O6, which is glucose. So over here, you can see there will be 12 NADP plus NADPH getting oxidized to 12 NADP plus 12 ATP breaking down to give 12 ADP. And over here, 6 ATP will break down to give 6 ADP. This is for 1 molecule of glucose. I'm showing 6 times because if 6 times it happens, how many carbons, how many ATP and all of that will be formed. Is it clear? Cool. We'll move on then. We'll talk about photorespiration. Anuj is a BSc student, okay. Samaira is asking how to cover the backlog, right? If you have a backlog, I suggest you don't watch live classes, you watch recordings, go in order, learn everything and then come to live class, okay? Cool. We'll learn about photorespiration now. I will disable the comments for a little while. How many of you know about photosynthesis? I'm only teaching right now. Cellular respiration, you might not know because that's the next chapter. So I'll tell you what photorespiration is. Photorespiration. Why are we calling it photorespiration? Because it's a little bit like photosynthesis and a little bit like respiration. That's why. How is it like photosynthesis? How is it like respiration? Let's see. So what happens during photosynthesis? This is my new favorite color. During photosynthesis, carbon dioxide plus water in the presence of sunlight will form glucose. It's not balanced, I know, and oxygen. Right? So basically, photosynthesis occurs in the presence of sunlight. That is the important point here. Next, what happens during cellular respiration? The exact opposite. During cellular respiration, you have glucose getting oxidized to form carbon dioxide plus water. Right? What happens during photosynthesis is carbon dioxide goes in, oxygen comes out. What happens during cellular respiration is oxygen goes in, carbon dioxide comes out. So this process called photorespiration takes place in some plants. In photorespiration, similar to photosynthesis, occurs in the presence of light and that's like photosynthesis. Like respiration, carbon dioxide is released. This is like respiration. So in this process, like respiration, carbon dioxide is released, carbon dioxide comes out. And like photosynthesis, this happens in the presence of light. That is why we call it photorespiration. So what exactly happens during photorespiration is, I told you about the enzyme Rubisco, right? Rubisco is an enzyme that will always on one end bind to RUBP. On the other active side, it can either bind to carbon dioxide or it can bind to oxygen. If it binds to carbon dioxide, Calvin cycle will happen. If it binds to oxygen, photorespiration will happen. So whenever Rubisco is showing its oxygenase activity, that is when photorespiration will occur. What is the significance of photorespiration? Is it beneficial to the plant at all? It is actually a very wasteful process. Okay. So during photorespiration, whatever carbon dioxide is fixed into organic molecules, that will break down and carbon dioxide will get released back into the atmosphere. Already fixed glucose, already fixed organic compounds, that will break down and carbon dioxide gets released. And to break it down, again ATP is utilized. So it's a wasteful process. And in order to, prevent photorespiration from happening, some plants have evolved into C4 pathway. Only in C3 plants you can find photorespiration. Otherwise, you cannot find photorespiration. So why is it so bad for the plant? Let's understand. First of all, let's talk about Rubisco. Rubisco, I'm sure everybody knows, is the most abundant enzyme in the whole biosphere, right? It is the most abundant enzyme. Now, this active site of Rubisco, if this is Rubisco, it will bind to RUBP here and it can bind either to carbon dioxide or to oxygen here. If it's binding to carbon dioxide, it's called RUBP's carboxylase activity. If it's binding to oxygen, we call it Rubisco's oxygenase activity. So how does this Rubisco decide what it has to bind to? It can either bind to oxygen or it can bind to carbon dioxide. How is it going to decide if it should bind to carbon dioxide or it should bind to oxygen? That basically depends on relative concentrations of carbon dioxide and oxygen around Rubisco. If around Rubisco there are nearly same amount of carbon dioxide and oxygen, let's say there are 100 molecules of carbon dioxide, 100 molecules of oxygen right around Rubisco. Then when they are in the nearly same quantities, or let's say 99 and 102, when they are nearly equal, at that time Rubisco will always have a greater preference for carbon dioxide. Okay, so it will prefer carbon dioxide more. It has a much greater affinity for carbon dioxide than for oxygen when their concentrations is nearly equal. Okay, let's say there is same amount of carbon dioxide and oxygen around it. It will obviously bind to carbon dioxide because it has a higher affinity to carbon dioxide. Now, during some circumstances, it so happens that more than carbon dioxide, the relative concentration of oxygen will be higher. This Rubisco enzyme cannot see and differentiate, okay, carbon dioxide is my friend, I have to bind with it, I don't like oxygen, it's wasteful for the plant. It does not know all that. These are random chemicals that are interacting with each other, right? So obviously, if around an enzyme that has an active site for oxygen and carbon dioxide, if around this enzyme the oxygen molecules are more, then the chances of this enzyme encountering oxygen is higher, right? Obviously, if oxygen molecules are 100 in number and carbon dioxide molecules are 10 or 12 in number, the chance of Rubisco coming across oxygen molecule or encountering an oxygen molecule is much higher. As a result, when carbon dioxide concentration is low, oxygen concentration is high comparatively, then it will show its oxygenase activity. Now what happens when it shows its oxygenase activity? You have RUBP. This RUBP has 5 carbons. If it binds with an oxygen, oxygen has no carbon, 0 carbon. It will form, see, if it binds with carbon dioxide, it will form 2 molecules of 3-phosphoglyceric acid. Over here, it will form 1 molecule of 3-PGA, which has 3 carbons, and another molecule of phosphoglycolate. This will have 2 carbons. This is bad for the plant. We don't need this. We have to eliminate this. This has to be changed into some other form. In order to change this phosphoglycolate into some other form, it goes through a series of reactions involving 3 different organelles: chloroplast, mitochondria, peroxisome. All of these organelles are used in converting this phosphoglycolate to something else. Okay, that is not part of your syllabus. Just understand. In that process, ATP is utilized. In that process, fixed carbon dioxide is released, which is very bad for the plant because fixing carbon dioxide takes a lot of effort. You saw that just now. Light reaction has to happen, ATP, NADPH has to form, that has to get used up, that will get used up. Energy is used up during carbon dioxide fixation. All of that happens when you fixed carbon dioxide in a plant, and then to eliminate this, that fixed carbon dioxide breaks up, oxygen gets released. ATP is used for a wasteful process. So, this is what happens during photorespiration. And in order to avoid this photorespiration, a wasteful process from happening, plants have evolved C4 pathway. Okay. So let's see what is on this slide. The most abundant enzyme. Its active site can bind to both carbon dioxide and oxygen. It has a greater affinity to carbon dioxide when they are nearly equal in concentration. Yeah, it depends on the relative concentration of oxygen and carbon dioxide. So whenever it's showing its carboxylase activity. When will it show its carboxylase activity? It will show carboxylase activity when there is high carbon dioxide to oxygen ratio. That means compared to oxygen, carbon dioxide concentration has to be high. Then RUBP will combine with carbon dioxide. This has 5 carbons, this has 1 carbon. It will form 2 molecules of a 3-carbon compound. This is called the C3 pathway. Over here ATP is also used, NADPH is also used. But okay, we're using ATP and NADPH. But we are synthesizing glucose, right, by utilizing ATP and NADPH. Over there, that will not happen. Over here, when does this occur? This occurs when there is low carbon dioxide, high oxygen. It will form a 3-carbon compound and a 2-carbon compound. This is called photorespiration. Over here, ATP is utilized. But sugar is not synthesized. Fixed sugar also, carbon dioxide will get released. So this is a wasteful process for the plant. Okay. Now let's understand, when does photorespiration occur in a plant? You know that plants get oxygen, carbon dioxide exchanged through the stomata. In plants that grow in tropical habitats where the sunlight is very harsh during some seasons, when the sunlight is very harsh, when the temperature is very high, when the air is very dry, the humidity is very low, in such conditions, transpiration will happen at a very high rate. You know what transpiration is, right? Water that is absorbed by the plant roots will be lost through the aerial parts in the form of vapor, through stomata usually. Okay, so that is called transpiration. So, if the plant is losing a lot of water that it's absorbing, then there is not sufficient water available for photosynthesis. So what the plants will do during those times when the sunlight is very harsh, air or the wind around it is very dry, when the temperature is very high, it will close its stomata shut. If the stomata is closing, what is happening? Transpiration rate is reducing. But if the stomata is closing, carbon dioxide from the atmosphere also can't enter the plant cells, right? Carbon dioxide entry is stopped. Carbon dioxide entry is stopped. Light is very harsh. Light reaction is happening. As an end product of light reaction, oxygen is forming. This oxygen is also not leaving outside. Carbon dioxide is not coming inside. Oxygen is not leaving inside. During that time, oxygen concentration will become much higher compared to carbon dioxide concentration. During that time, this process will take place. Alright. So that is about photorespiration. If anybody has any doubts, you can ask me. Srijana, please post your doubt right here. Is it the reason old people prefer not to sleep under tree at night? I don't know. Actually, this stomata will be shut at night. Okay. In most plants, unless they are camp plants, stomata will close at night. It will open only during the day. Maximum, most stomata are open during the peak afternoon. Towards evening, it will close shut. Technically, it should not be a problem, but yeah, that is the reason. Anaerobic respiration occurs in muscles. Kusma, we are not even talking about anaerobic respiration yet. This chapter is about photosynthesis. About respiration, we'll talk in the next class. Okay, somebody wants carboxylation step explanation. Pratik. Okay. So, Pratik, what happens during carboxylation is you have a 5-carbon compound which is called RUBP. This 5-carbon compound will pick up another carbon from carbon dioxide, and that step is called carboxylation. As a product of carboxylation, instead of getting one 6-carbon containing molecule, we get 2 molecules each containing 3 carbons, and that is catalyzed by the enzyme Rubisco. Okay, so I will ask you some questions. No chatting in the chat box. Okay, please pay attention to what I'm saying, otherwise I will turn it off. Now I will give you some questions. Start answering them quickly. PYQs we'll discuss in the last class of this chapter. I want you to come prepared for that. On Wednesday, we'll discuss PYQs. Tomorrow, we will finish this chapter. So, whatever has been covered so far, please be thorough with it. I will be disappointed if you all don't get 100% correct answers. Okay. So, I will ask some questions. Which photosystem is found in the stroma lamellae? Only. It's PS1. ATP synthesis occurs only in cyclic photophosphorylation. Is it true or false? No. ATP synthesis occurs during non-cyclic as well, so it is false. Proton concentration is greater in the lumen compared to the stroma. This is true. Proton pump in photophosphorylation. Which acts as the proton pump? It is the cytochrome B6F complex. Site of photolysis of water. Where does water splitting reaction happen? It happens towards the lumen side of the chloroplast. First stable product of C3 pathway is 3-phosphoglyceric acid. The first stable product of C4 pathway is oxaloacetic acid. This contains 3 carbons and this contains 4 carbons. Next, primary acceptor in C3 pathway is RUBP. The name of the enzyme that catalyzes carboxylation in C3 cycle is Rubisco. 10th question, where does NADPH form during non-cyclic photophosphorylation? It happens towards the stroma side of the chloroplast. How many of you got all correct? Many of you answered correctly. Divya got confused in the second one. Divya Mishra. Cool. Some more questions? Which part of ATP synthase forms a transmembrane channel? It is the CF0 particle. 2-carbon compound formed during photorespiration. Phosphoglycolate. During photorespiration. 13th question. What is the correct answer? ATP is utilized. Option C is the correct answer here. Carbon dioxide is released. ATP is utilized. Oxygen is not released. Glucose is not synthesized. During photorespiration, Rubisco shows which activity? It shows oxygenase activity. Number of ATP used per turn of Calvin cycle? 3. Has anybody answered? I want you to read the 16th question carefully one more time and answer again. Kirtana, you've made a mistake. Does NADPH get produced during Calvin cycle? Does NADPH get produced or does it get oxidized? Does it get used up? It doesn't get produced. It gets used up. So it's 0. Correct, Aisha. For those of you who are asking for poll questions, we will discuss poll questions on Wednesday. I want you all to come prepared. Sahiti has got everything correct. Congrats. Okay. So those of you who got questions incorrectly, that shows where you have not understood properly. So go back and revise. I want you all to study whatever is taught today itself. Today no poll question, Prasanna. Okay, please study. Come well prepared. We'll solve a lot of questions on Wednesday. Alright? So that's all for today. I'll see you tomorrow at 12 o'clock. Take care. Bye-bye.